From Microbe TV, this is TWiP, This Week in Parasitism, episode 124, recorded on January 3rd, 2017. Joining me today here in New York City, my two parasite colleagues. They are parasites. We all are parasites. Dickson Despommier and Daniel Griffin. Welcome to 2017.
Hello, everybody.
Hello, everybody. Happy New Year. Welcome to the new year.
Yep. This is the first TWiP of the new year, but it's also the first Microbe TV show.
No kidding.
Yeah.
Okay.
You know, it's the first one.
Get to kick it off.
January 3rd. Thank you very much.
What does that mean when you say it's the first Microbe?
Well, you know, I do all these other podcasts. Right?
Yes.
TWiP, TWIM, TWevo, etc.
And this is the first
Urban agriculture
of the Twix universe.
That's not going to be done until September.
Stop it.
That one.
It's harvest time. Come on.
And I want to thank everyone for their birthday wishes. Thank you, Daniel. Thank you, Dickson.
You're welcome.
Yesterday was my birthday.
Yesterday was his birthday.
January 2nd.
That's fantastic.
And you said in digital language you are?
I am 1 followed by 6 zeros.
That's perfect. That is absolutely perfect.
Binary for 64.
That's fantastic.
Oh,
it's a great number.
There's a great song about
There is a great song. Will you still need me? Will you still feed me? Yeah,
that's right.
When I am 1 million. It doesn't rhyme.
That's right.
Dixon is a different number in binary.
He's a different, much different number.
I have arrived at the song. But, you know, 64 is the new 40.
Is that what they say?
Yeah.
I hope that's true for you.
Well, you look quite young for 64. I would have never pegged. I would have said 54. Maybe.
I hope that works out for you.
Thank you very much.
As they would say.
I hope
it didn't for me, but
64.
Well, no, 64 was pretty good actually. The early 70s weren't so bad either, but
your early 70s or the early 70s?
My early 70s. Okay. But you know, the closer you get to 80. Mm-hmm. The less like 60 you behave. That's all I can say. You get slower.
When I was very young, when you turned 60, people didn't like it because you had to retire. And
that's right.
Now people keep going until their 80s.
Exactly.
All right, let's start with a case from TWiP 123. Daniel, that was 2 weeks ago, right?
Yes, so we've been doing pretty well, keeping on schedule here. And let me, as I like to say, remind everybody or to acquaint those who are tuning in for the first time.
Right.
We discussed the case of a nurse in her early 20s. She was a recent graduate from nursing school, and she had decided to spend a year doing a global health internship in the Dominican Republic on the Haitian border, and she had developed an issue on her foot. It was a skin issue. Initially, she was told it was fungus, but it was not improving with topical antifungal cream, actually getting worse, as she says. Been going on for several days, only 1 foot. She's healthy, no past medical, surgical allergies, no medicines, HIV negative. She's living with a local family in this small village. The family has a daughter, a wife, a husband, and a cat who I think we mentioned 1 month prior had kittens. No toxic habits are reported. Originally from the U.S., she swims in the local river. She walks barefoot to and from. She sometimes has her shoes off in the house. She eats the local food, lots of exposure to dogs, cats. And she describes this area on the foot as very itchy, but it's not open. There are blistery, a couple of blisters, and this rash is described as serpiginous. So it's an irregular, raised red line on her foot.
Wasn't this the second consecutive case involving cats?
Cats were mentioned.
Well, cats were mentioned, I should say. The previous case as well, right? Cats were mentioned.
Yeah. Cats were mentioned.
The Dutch lady, right?
That's true. Okay. That's true. The mango eater, as we affectionately refer to her.
Despite this being the holidays, we did have guesses.
We did.
First one is from David, who writes, "Dear hosts." That's a little play on words.
Yeah, we got it.
We got it, Dickson?
We got it.
It was extremely interesting to hear all of the different diagnoses in the previous podcast. I think you had most of us stumped but the listeners of TWiP are quite erudite, and all of the guesses were well thought out. In the case of the nurse that Dr. Griffin examined, I believe her foot has been invaded by a hookworm species, most likely Ancylostoma brasiliense, which is causing the itchy, raised, serpiginous rash on her foot. This zoonotic disease occurs when a dog or cat hookworm accidentally parasitizes a human host, which is not the parasite's definitive host. The worm aimlessly wanders throughout the dermal tissue, leaving a trail of secretions containing hydrolytic enzymes that trigger an inflammatory reaction and produce a creeping eruption characteristic of cutaneous Larval Migraines. Treatment of this condition are numerous and include oral treatments such as albendazole 1+ dose and ivermectin 1 dose, topical applications such as thiabendazole 10 to 15% 3 times a day for 5 to 7 days, topical freezing agents such as liquid nitrogen, not recommended. All things considered. Wasn't that the name of a radio?
NPR show. Yeah, it's a very good one.
I'd like to think the theme revolves around worms of all shapes, sizes, and species. We have seen an arthropod worm, maggot, and then moved on to a nematode hookworm. I wonder if the next case study will showcase a tapeworm or a flatworm. Perhaps an alternative recurring theme is young professionals traveling and contracting worms or any illness for that matter. Thanks again for the informative podcast. Sincerely, David P.
Right.
Dickson.
Peter writes, "Hi, TWiP team. The serpiginous lesion that changes location clearly points to cutaneous larval migraines, or CLM, described in Parasitic Diseases 6th edition under aberrant nematode infections. This is the result of a zoonotic hookworm infection from either Ancylostoma brasiliense or Uncineria stenocephala. These nematodes parasitize dogs and cats. There are other nematodes that cause CLM, but they do not always produce serpiginous skin lesions. CLM can be diagnosed from the characteristic skin lesions. Biopsy is not recommended. Cutaneous larval migraines is self-limiting, as the larvae are unable to complete their life cycle in humans. According to the CDC, migrating larvae usually die after 5 to 6 weeks, but try to live that long with it without trying to do something about it, and you'll find out that it probably lives a lot shorter time than that one. According to Parasitic Diseases, 6th edition, migrating larvae can survive in the epidermis for about 10 days. Albendazole and ivermectin are both effective treatments, but according to the CDC, ivermectin is not approved for this condition. Secondary bacterial skin infection may need treatment with antibiotics. Antihistamines and topical corticosteroids can be added to the treatment to help pruritus, or to help resolve pruritus, because it doesn't actually help pruritus. Cutaneous larval migraines was also the parasite in the case study for TWiP 95. It has also been covered on Monsters Inside Me, and those are good references as well, and he lists a couple of links to the Oxford Journal and the CDC travel guide. Regards.
Thank you, Daniel.
Ruben writes, "Dear prolific podcast shedding hosts, as one of your many infected recipients, I have to free myself from the itch to share an educated guess. It comes straight from the book of Dickson and Daniel, plus other apostles of parasitism. The unfortunate nurse recklessly walking barefoot on the feces of dogs and cats may have a case of cutaneous larva migraines caused by larvae of the dog and cat hookworms, Ancylostoma brasiliensis or Uncineria stenocephala. A. brasiliensis, it appears to be more likely. Now the treatment, hit the larvae with albendazole and ivermectin. Cover the larvae with thiabendazole cream or freeze them if you can guess where they are hiding now. In conclusion, sometimes the ignorance, a frequent companion of youth, is bliss, but not in this case. Please put your shoes on."
Right.
With kind regards, Ruben from La Jolla, California. And right. P.S. This sentence in your online description of case study for TWiP 123 needs fixing. Oh, it says East's local food exposure,
right.
dog, cat, and sister.
While someone is
you.
reading subsequently, I shall fix it. Yosef writes, "My main differential for this case would be a worm that is crawling through this young woman's foot, leading to cutaneous larval migraines, a.k.a. ground itch or plumber's itch. My most likely guess As to the culprit would be Ancylostoma brasiliense. This worm normally parasitizes cats or dogs, but can accidentally get into us. Once in us, they cannot complete their life cycle, since they lack a collagenase enzyme necessary to pass through the basement membrane in humans. Thus, they squirm around in a futile attempt to find the correct blood vessel until they die. The immune reaction by the host leaves the serpiginous lesion. However, I've been told that the lesion no longer houses the worm, but rather only shows where the worm has been. If she starts to develop pulmonary symptoms, GI symptoms, positive hemocult, or symptoms of iron deficiency anemia, then I would be concerned about other worms that are more successful in humans. Worms such as Ancylostoma duodenale and Necator americanus and Strongyloides stercoralis all have the necessary collagenase enzyme and can travel through the outer layers of skin, make it into a blood vessel, reach a pulmonary capillary, burst through, travel up the trachea, and finally be swallowed into the GI tract. Wow. Yep. What a trip.
What a trip.
I don't think I would even test this woman for the particular worm, since the treatment would be ivermectin in any case. I predict that she got better, and I hope that the knowledge of having worms in her body does not deter her from future global health adventures. Sincerely, Yosef. And I might add in a postscript, the knowledge of having worms in her body compels her to wear shoes in the future. Right? What do you think of that?
It could happen.
But youth, they don't always
often listen.
They don't often listen. Dickson, can you please take the next one?
I will attempt. Tony writes, Dear Doctors, I am Tony, the microbiologist from Spain, writing again. As always, here is another interesting case study. The main symptom is the presence of itchy, papular, elevated lesions on one foot, so the differential diagnosis could be 1. A fungal infection. It is usually itchy. The unresponsiveness doesn't rule out a fungal infection, since there are several highly resistant species, but makes it less likely. 2. Another option could be scabies. It produces an uncontrollable itchy feeling, and on inspection we can find the tiny burrows on the skin as 1 centimeter long whitish lines. The location is not usual for scabies, and given its height transmissibility, we must find more people suffering the same type of symptoms. 3. Another option could be tungiasis, caused by Tunga penetrans. This infection is acquired walking barefoot on humid soils, as is the case in our patient. The dermatologic sign is a less than 1 centimeter wide white patch with a black dot in the center and can be itchy, burning, or painful. 4. Lastly, another option, which is my best guess, could be cutaneous larval migraines, a term used to describe several types of parasitic infections that can cause migrating lesions in their immature stages. Several parasites can cause larval migraines. Toxocara canis, Toxocara cati, both acquired by ingesting infected larvae. Fasciola hepatica, ingesting freshwater vegetables like watercress. Strongyloides stercoralis, skin penetration of infective larvae. Gnathostomiasis, among others. The most usual etiology for larval migraines is A. brasiliense, A. caninum, Uncenaria stenocephala, collectively known as zoonotic hookworms. In the Americas, the most common species is A. brasiliense. These are the equivalent in dogs to human hookworm. The immature larvae cannot penetrate the skin, so they are unable to finish their life cycle as they do in their definitive hosts. The speed at which the larva moves is also important. In the case of zoonotic hookworms, the speed ranges between 1 and 5 centimeters a day. Strongyloides is much faster, with a 5 to 15 centimeter per hour. That said, the speed is important. We can measure the speed just by putting a mark on the skin of the moving front and another mark 1 hour later. P.S. Do you believe in coincidences? I want to share with you my experience. In 2005, I was working with my wife in southern Mozambique with a non-NGO involved in a health project. It happened that my wife began to feel uncomfortable for several days, and someone suggested to do a blood test to check for malaria. It then seemed unlikely since she was taking preventative pills, mefloquine. So finally I did the thin smear. That exact day was her birthday, the 25th of April. To our surprise, I diagnosed her with a 2+ infection with Plasmodium falciparum, and it also resulted in the 25th of April. It also is the result that the 25th of April is the World Malaria Day. Now, that's coincidence. Thank you very much, and happy Christmas for everybody. I hope many more fantastic episodes for 2017. That is a coincidence.
Yeah, I mean...
What better day in the world to catch malaria, though, than on Malaria Day?
You could get polio on World Polio Day. You could get AIDS on World AIDS. You could get rabies, et cetera, et cetera.
Lord.
It goes on and on.
You'd never make it through the year.
No, you wouldn't. Daniel.
Yeah, I was just thinking, like, what is it, 500 million cases of malaria a year, so 1 million plus people get malaria on Malaria Day.
That's right.
That's very bad, isn't it?
Yeah, no,
that is right.
And you were wincing at some point during the reading. Was there anything that bothered you, Dickson? We'll go back? Okay. We'll do that. Okay. All right, John writes, "Hi, three wise men. Well, 'tis the season." Okay. I'm the Zoroastrian in the group. Just if we're going to choose which
we had
wise
to judge
men
by your hirsuteness, I would say no, but that's
perhaps not. Okay, I have been listening for a while. This is my first email and first case study guess. My background is in software, but my research for this case involved Parasitic Diseases 6th edition and Wikipedia. Who needs Wikipedia, right? When
exactly
you've got
right.
My initial guess, mainly because it's one of the few parasites I knew about, was the guinea worm, Dracunculus medinensis. It affects feet and can be ingested by swallowing infected copepods, by swimming in infected water, as this patient did. However, it is now rare due to significant eradication efforts, including by President Carter, and occurs mainly, maybe even exclusively, in Africa, not Central America. Also, infection with worm is usually associated with intense pain when the blisters have formed, but the patient described it as itchy. I briefly considered Strongyloides stercoralis, but it didn't really fit. My actual guess is cutaneous larva migrans caused by the larvae of cat or dog hookworms. It seems to fit for several reasons. It's common in the Caribbean. There is a cat in the house. She walks barefoot. The woman walks barefoot. It presents most often in the lower limbs or feet. It is itchy. The lines on her feet are serpiginous. Thanks, Dickson. It moves. There are small fluid-filled blisters. For treatment, I would refer you to Parasitic Diseases 6th edition, page 313 through 314.
Who could pass that up?
By the way, Dickson suggested during the episode that the chemical elements have been around for the 14 billion year history of the universe. Now, this is true for hydrogen, about 73% of normal matter, and helium, about 25% of normal matter. A tiny amount of lithium and beryllium and maybe boron were formed during the same period, within 20 minutes of the Big Bang. Of the remaining 2%, most, up to iron, were formed later in stars by stellar nucleosynthesis, or, for elements larger than iron, in supernova by supernova nucleosynthesis, in processes that are still going on today. Listener pick, if you're interested in stars, galaxies, and the universe, Professor Pog of Ohio State University has audio recordings and lecture notes for his An Introduction to Solar System Astronomy, Astronomy 161, Life in the Universe, Astronomy 141, and Introduction to Stars, Galaxies, and the Universe, Astronomy 162 courses. Regards, and thanks for all the podcasts. Best of luck for 2017, John from Limerick, Ireland.
Right. So, Dickson, remember that not all the elements were present the next time you have the opportunity.
I will. The next time that comes up in a cocktail party, I'll be sure to give the correct answers. But I'm sure within the first million years, most of them were created, don't you think?
No, it says later. And,
how later?
And processes that are still going on today.
Yeah, I don't doubt that. But, okay.
I think quite a bit later,
I mean,
actually.
You said
they sort of talk about the initial formation, and then it takes a while for the things to sort of coalesce and concrete.
You know, we have this web space telescope that we're about to launch. We'll be able to look back almost to that time to see if there were galaxies a million years.
Why don't we get
after
Neil deGrasse Tyson on this podcast?
I would love to get him on it.
And have him talk about it.
He would be great.
Of course he would. He's right here in New York City,
He might even like to talk about parasites. We don't know.
Didn't
you one time cancel on us because you had to go hang out with
I was supposed
Bill Nye,
to go
the
on
science
his show.
guy? And
so, Neil's show was being hosted by Bill Nye, and they had invited me to go down and talk about Ebola. So I said, I can't do TWiP. And then they canceled it, so we did TWiP after all, I think. Yeah. And they said, we'll have you back, but they never asked.
No.
Yeah, so well. Mike writes, "Hi guys. At first I was thinking it might be swimmer's itch, schistosomiasis, but now I'm leaning towards plumber's itch, i.e. hookworm, ancylostoma duodenale, or Nicator americanus. I still remember Dickson telling a wonderful story of hookworm in TWiP 22. People, remember to make your outhouse at least 6 feet deep. I used to have a micrograph of hookworm as my desktop image until my wife saw it and freaked out." Mike in Oregon.
Way to go, Mike.
Oregon. Dickson.
Sathish writes, "Dear TWiP parasites, the curious case of the snake-like rashes on the nurse intern sounds like the classic textbook description of cutaneous larval migraines caused by the third-stage larvae of Ancylostoma brasiliensis, which burrow into the skin. Given her history of barefoot adventures, it is easy to imagine the repeated exposure to this parasite abundant in soil. The disease is also known as creeping eruption, with most cases appearing on the feet with the serpiginous lesions up to 5 centimeters long. It can also be present in the buttocks, abdomen, legs, arms, and face. It is usually self-limiting, but a short course of oral albendazole or ivermectin can be used. P.S. I'm currently working through parasitology as part of my master's in infectious diseases from the London School of Hygiene and Tropical Medicine. I am grateful to TWiP, especially your initial 50 episodes, which are a great resource to understand the basics of parasitology and helped me bridge the gap between the bench and the bedside. In fact, it was a sweet coincidence that I was just beginning to read a hookworm chapter in my textbook when I was wondering about this case. The book chapter had all the key words, moving lesion, serpiginous, which were dropped quite smartly by Dr. Racaniello into the podcast, which got me hooked into the chapter. The final giveaway for me was a picture of the classic lesion, which was referenced to Despommier et al. And Parasitic Diseases 4th edition. Aha! I never looked elsewhere from that point. Dr. Griffin, I remember your saying that you visit India frequently. I was wondering if I could get an opportunity to meet you face-to-face when you come here for your talks. I have grown so accustomed to all your voices, jokes, and insights. I learn a lot from you and feel a certain connection, dare I say emotionally, with you all. I guess TWiP is like a boy band, and your talk is music to us all. So please let me know when you come to India the next time so I can visit you and get my parasitology books autographed. I live close to Vellore in South India. I wish you all a happy New Year 2017.
Yep. Do you ever go close to Vellore?
Last time I was actually in Vellore in, November? Yeah.
Right. And I go over there.
No, it was last November. It was November of 2016.
Yeah.
So just a little over a year ago. Yep. So we will, we could announce on our podcast. And
we will keep in touch. Don't worry.
Stay tuned.
Stay tuned. That's the way to put it.
All right, Daniel. What was the outcome?
So do you think there's a consensus?
What were you cringing at in that one letter?
I was cringing because a couple of these diagnoses were a little bit off. For instance, strongyloides stercoralis doesn't cause serpiginous lesions, and because they're so fast to penetrate that you wouldn't even notice the lesions unless you were penetrated more than one time. 2 or 3 episodes of infection can lead to a hypersensitivity state, which then shows you each larva where that penetrated. You get this little red papule. No. But otherwise, it's pretty innocuous. There was one other thing, too, I wanted to say. This woman that was walking barefoot through the cat and dog feces, you don't have to actually do that because, remember, hookworm, when they hatch from the eggs in the feces, they can crawl. So they don't have to be where the feces is. And I think anybody that's looking down at their feet when they walk back from the beach wouldn't step in dog feces necessarily. But it wouldn't matter. If you step within 2 feet of it, you're still going to be subjected to the worms. So I would...
Isn't that sort of frightening?
Caution against...
You're in the house and you see like, oh, there's some cat stuff over there. But meanwhile, unbeknownst to you, there are these larvae that are squirming across the floor towards you.
Streaming out from the feces.
Yes.
Right. But I wouldn't say this happens in the house, mostly in the soil. Because in the house, the dry floor would probably kill them. I think they would die from dehydration. But in the soil, because there is moisture and capillary action of moisture in the tropics, at least, the worms do very well under those conditions.
Dickson, how many worms per gram of feces would there be?
It depends on the heaviness of the infection. Now Ancylostoma produces about 3,000 to 5,000 eggs a day, as I recall, but that's just a guess. I'd have to go back and look at the book to make sure.
I think you're actually right on that.
And so 3,000 to 5,000 eggs a day per adult female. What if you had 10 female worms or 100 female worms? In that case, you multiply times, and then you get a really lot of larvae coming out of those fecal deposits. And that's every day for about 3 to 5 years. So these dogs can remain infected for quite a while. Yeah, it's a serious dermatologic condition that we see occasionally, and it's associated with people at this time of the year returning from tropical locations where there's no control of animals on the beaches. Right? So like Puerto Rico, for instance.
Sure.
Puerto Rico has laws against this, except for one place, believe it or not.
Laws against what? What are you not allowed to do?
Pet animals down to the beach. You're not allowed to run your animals on the beach. And they did that on purpose because they've known about this for a long time. However, if you own your own beach, you can do whatever you want. So all the beaches are public in San Juan except the Hilton. The Hilton actually built their beach so that you can do whatever you want there. You can bring your pet dogs or cats there if you want. And as a result, there is the opportunity for encountering larvae.
On the Hilton Beach.
On the Hilton Beach, right.
Wow.
Daniel, what'd you do with this young lady?
The nice thing, and I think, compliment all our listeners who wrote in, is that this is a clinical.
Good job.
This is a clinical diagnosis, and people, I think, cinched it there. There's no tests that we're going to do in these people. It's going to be this clinical presentation of a serpiginous, motile, moving lesion, and it is interesting. It's going to get better on its own, but 10 days later, it's going to get better on its own. So this worm or worms, depending, this worm larva is going to continue to migrate around your foot. So in this case, I was asked about this. I saw it, and this is a place, I think we mentioned that electricity is not always available. It's raining and overcast. The lighting is not very good. So I actually used my iPhone to take a nice high contrast photo of her foot. And then it became very apparent that, it really had the pattern of this serpiginous, serpent-like raised end. So, immediately I recognized this and suggested that she take medicine for this. Now, she was not able to get the medicine until the next day. She got it from the hospital pharmacy. And in that period of time from the diagnosis till the next day, the worm started to migrate proximally up the foot. And but then she took the medicine, killed the worm, and over a period of time, it.
So this is a case where the clinical presentation is so unique that you don't need to do anything else.
It's true, and they recommend not. I mean, you don't want to be cutting into somebody, or you just
that's right.
You treat them.
It's not a surgical procedure to get rid of.
You're going to do more harm, right? Than freezing or cutting or all these other things. So,
so what did you give her?
She got albendazole.
Yeah. Well tolerated.
Well tolerated. And it's really amazing. Here in the U.S., that would be like a ridiculously expensive option.
That's
which is really interesting, right?
Yeah, this is crazy. To see how this drug has skyrocketed just for domestic use in the United States is just absurd.
Yep. So
it's really cheap to
with her, I did 400 milligrams, just one pill a day for 3 days, which is plenty.
Yeah.
As we talked about, she's going to get better if you do nothing, but at least you're going to stop it from crawling around, and it's itchy, and it's particularly now you realize, oh my gosh, there's a worm crawling around in my skin. Yeah. It's not an exciting prospect. And yet in the U.S., it's a couple $100 a pill, and the pills are 200 milligrams. So you're talking like $400, you're talking $1,000 to treat this in the U.S., where down in the Dominican Republic, you're talking like 30 cents.
So you're a physician. We're not. Tell us what's going on here.
As far as the drug prices.
Yeah.
Well, we're all in cahoots, and we get a big. No, it's sort of amazing.
Why is there such a discrepancy?
Well, it sort of amazes me, and it's the unfortunately
it's greed.
Would you say it's greed? I wouldn't use that word. I think it's too I think it's too simplistic. I mean, everybody likes, but I think what it is that we allow the capitalist market to sort of have free range, in our healthcare system. And there's a couple things. Someone famous once talked about one of the things that does not do well in a pure capitalist model, and healthcare being one of them. Because when you're sick or have an illness, healthcare is not a luxury item,
Right.
Much like food or a number of other things. So there's a certain situation where, as a community, as a nation, as a people, you sort of want to be working together. And it's fine if people are innovative and hardworking for them to make a good living. But when you're sort of limiting medicines, it's a little bit tricky.
Yeah.
So I think that's what it is. I think we probably need to introduce some sort of balance that allows companies that are bringing new medicines to the market to do well, but at the same time not at the expense. So it's a tough balance to achieve. And in places like Dominican Republic, they're not going to make any money. You just can't make money off poor people. And if you are making money off poor people, then there's a pretty serious problem there.
That's right.
You can make money off wealthier people. I guess that's what they're trying to do in the U.S. But yeah, I think the balance has been lost.
The drug wasn't always that expensive in this country. When it first came out, it was much cheaper than that. So it reminds me of the story with the EpiPen.
I no longer carry one just because it's too expensive.
Wait a minute, that's not what I said.
I mean, my severe bee allergy, I've just got my fingers crossed.
No, don't do it. Don't do it. You should carry one, but it's hard to resist that temptation to fill your coffers with as much as you can, because it is expensive to develop drugs, and lots of drugs fail and they don't make it to market, or they do, and then there are side effects that are discovered later, and then they have to withdraw the drug, and they've spent a ton of money getting it out there. So the drugs that do work, that don't have these problems, are looked at as their cash cows. And I knew a lot of people at Merck, and I know you did too, Vincent, because your wife worked over there for a long time, and we've known the ivermectin story, and I know the thiabendazole story, and mebendazole too. Those drugs went through a series of screens, and lots of people were involved, and lots of research, and lots of trials, and you add up all that money, and somehow you have to recover it. And it's too bad that there's no compassionate use committee at the governmental level to set aside certain drugs that could be used, like this one, for instance, because I think this is, it deserves a different price range than it's got.
Yeah. No, I think it falls on us. I mean, we often victimize or go after like big pharma, big drug companies, but it's our system. In the United States, the government is us, and so it's our system that creates this. Yeah, you're right. But to get back to the science, one of the things that's interesting, right, is how many people know, just in general, who may not listen to TWiP, that it's unsafe to walk around barefoot in the third world, right?
Correct.
Mm-hmm. Or that, when you wash your shirt in certain parts of the world, you can't just hang it on the line to dry, right? I mean, there's a lot of this stuff
that's true.
People go into these situations. You grow up in the U.S. and, you're going to go swim in the river, you walk around barefoot. You're going to eat a mango. You go ahead and just take a big bite out of the mango. There's a lot of these things that people just don't know. And then they go to these places, they learn for the first time, oh my gosh, I've got a flea living in my big toe. I've got maggots from that mango I just ate. Right. I've got a zoonotic hookworm crawling around in my foot because I can't walk barefoot in this country. And so
early on in our history, I mean, I agree with you entirely, except at the turn of the century during the Hookworm Commission in this country. Mm-hmm. That was the biggest problem, and that's not creeping eruption. That's hookworm disease, because that was the human hookworm we're talking about.
The proper collagenase, it goes where it wants to go.
It sure does, and so the history of forcing people to wear shoes, it became a law that you had to wear shoes, and then people rebelled against that law. By the way, this guy Bailey Ashford in Puerto Rico, working for the United States Medical Service, was responsible for that law being enacted in Puerto Rico. And he's a hero, because actually what he did was he essentially, through public health measures, wiped out hookworm disease. But at the same time, he employed some what we would consider draconian measures to get that done, because he required compliance by everybody in order to make it happen. What was also needed was a good drug, and we didn't have a good drug for a long time, and then all of a sudden we had a wonderful drug. So it's a long history. So we've evolved past that. Isn't that interesting? Because it's like going back into a book about genetics and finding the word DNA for the first time as possibly the genetic material. And then it became the Watson and Crick model. And now you don't even see that anymore. You just see DNA again.
Yep.
And now you don't see shoes any longer because you can walk wherever you want in the United States. But if you go somewhere else, you might be in something other than deep yogurt.
So maybe we should review. I think we mentioned we're going to try to review the life cycle on these, or get people.
Yeah, I think we should do that.
So, because that's one of the interesting things. So, the hookworm, and a lot of people think that you get sick, you get worms from stuff you eat, but in this case, it's actually, in her case, not something she ate. It was the fact that she was walking around barefoot.
So we should just mention briefly all the parasites that are capable of penetrating the unbroken skin. Okay, so we have the schistosomes, the Cercaria that swim in water, in fresh water only, and they will penetrate a hair follicle, and that's how they initiate an infection. And then we have strongyloides, which penetrates the unbroken skin, but you can also swallow it, and it will penetrate the unbroken skin of your small intestine. And then, of course, we've got the hookworms that can penetrate unbroken skin as well. So those are the ones that we have to be on the red alert for.
So these, in this story, were fecal contamination from dogs and cats.
That's right.
Those are the natural hosts?
For this particular species.
Right,
yes,
for this species.
Right. For Ancylostoma brasiliensis.
And once it's in humans, it's dead end, doesn't go anywhere else.
That's right. That's correct. That is right.
But Ancylostoma
duodenale
and
Necator americanus, those
are the correct ones.
And they have them down there too. Don't deny that fact at all, because people do randomly defecate in places as well.
Right.
And that helps to keep this infection alive.
Now, we would be excreting worms.
Eggs.
They would hatch, and then they would get into someone else.
That's right.
How?
By them stepping on it.
Same
or
thing.
sitting down on the grass. Or plumber's itch. They mentioned a word that I hadn't seen in a long time called plumber's itch. In the South, a lot of houses are raised off the ground to keep a cool air mass underneath the house, to keep the air. So the dogs all sleep underneath there because it's nice and cool, right? Yeah. So what happens when something goes wrong with the plumbing? Well, the plumber is called in, right? The plumber, he takes his shirt off.
Oh, my God.
And then he grabs a hold of a pipe and he slides up underneath the building. And I used to say to the students, he could slide because it's lubricated with a thin film of dog feces all the way out to where the pipes are.
This is terrible.
And they would come back, and their backs would be absolutely, it looked like a Rand and McNally road map of some congested northeastern corridor place. And those were just larvae like crazy penetrating through their backs. And they would drive them crazy because they obviously didn't know what was wrong in the beginning, but now they do. Imagine being penetrated by thousands of these worms.
Now, how do the dogs and cats transmit it to each other? By eating feces?
No, they step on them also.
Oh, their little foot pads can take up worms? Yeah,
absolutely. Yeah, it's the same cycle in dogs and cats as it is in us.
And you can't tell them to wear shoes.
You could, but they wouldn't listen.
Maybe you could. Like I've seen those northern dogs, right? They wear the little booties. That's what we're going to have a campaign to get all the dogs and cats in the DR. And they have little booties.
That will work.
We're going to put our logo on there.
If Bailey Ashford were still alive, he might be able to pull that one off. But I don't think the rest of us would be capable of that one.
But you got to start when they're kittens, right? When they get
he had the military behind him. I'm not kidding. It was
all right,
military.
I believe you.
Yeah.
All right, are we done?
I don't know.
No, I think those are the big. I think those are the big things. So the eggs are going to come out in the stool. They're going to need to go through a sort of a embryonation. They're going to hatch. The larvae will, as we mentioned, they'll actually quest. They have this. They'll climb up grass stalks. They'll wander around the floor. They're
quite behavior oriented.
And then they'll actually enter often through the hair follicle. And then, as some of our emailers wrote, some of these lack, the difference between the aberrant ones and the non-aberrant ones, sort of our hookworms, are having the right enzymes to break through the basement membrane and migrate around as they need be.
So the question is, if you were to inject these larvae intravenously instead as an experiment, would they complete the life cycle in humans? And so there is one parasite that lives in Australia, which is an Ancylostoma of dogs. It's a different species, and I'm blocking on the name of it right now, but some human cases have been detected. So occasionally there is a breakthrough infection. And it doesn't mean that there's going to be an epidemic of this in people, but it does mean that people are occasionally susceptible because
Yes.
the larvae do find their way to the right place.
All right.
Okay.
Let's move on to a paper.
Good going,
What do you
by
think?
the way.
Should we
Good
do a
going.
paper?
Oh, absolutely.
Oh, should I mention the theme? Because we're going to break with the theme. I should probably mention.
Oh, that was it.
before.
Just was a 2-episode theme.
Well, I was going to go for a while, and then I said, you know what? Let's throw a little variety in here. So I was starting along the theme, and we're gonna bounce back to this, but of healthcare workers who go to these areas, like someone guessed that, and then end up getting sick themselves. And I think that's an interesting. And I've got a few more I'll sort of pepper in here, and I'll mention that they're along this
interesting,
theme.
though, is you have never gotten sick in all your travels, have you?
I don't get sick anymore.
Well, you do, yeah.
Well,
I
it's 'cause you're thoroughly
used to
immune.
get horribly sick.
I
Like
see.
I remember as a young man in Mexico, and this subsistence farmer wanted us to drink his agua bueno, and I turned to my friend. And I was like, listen, we're either going to get sick or we're going to be rude, and we're like, let's be polite
Let's go
and
get
sick. And horrible Giardi when I was working in Kathmandu. And so, I mean, I used to just think every time I traveled, I would get sick. And now I don't get sick anymore.
Is that because you're careful?
No, not really.
You're not really careful?
Not.
no. He
Barely not. Well, no, I
Daniel
followed
was
your
done.
advice. Like when I was swimming in Lake Malawi, I toweled off vigorously before the cercaria could penetrate my skin.
I wouldn't have been swimming in Lake Malawi to begin with, but that's another story.
You wouldn't, Dickson?
No, I would just. You know what I would do? I would get into a boat
Yeah.
and get rowed out into the middle of the lake, and that's where I would swim, because the snails don't live in the middle of the lake; they live along the edge of the water. So that's where the cercaria will be found. And so if you go out about 100 yards and then start swimming, you're fine. The only thing you have to worry about there is the Nile crocodile.
Yeah, that's what I was worried about. Yeah, I had a buddy who went out fishing, and and down went the boat, and he came very quickly swimming toward shore because there's crocodiles all in that water. So that's mainly what I worried about.
Right.
All right, we have a good paper, right?
Plus neglected tropical diseases. Replication protein A presents canonical functions and is also involved in the differentiation capacity of Trypanosoma cruzi. This is from a group of individuals, mainly from Brazil, mostly São Paulo, but one from Minas Gerais. And it's all about T. cruzi, which is the agent of Chagas disease. Would that be correct?
That would be exactly correct.
Could you tell us the life cycle, please, Dick?
I would briefly summarize.
Briefly.
I will.
That's it.
Actually, it's
Are there any
really
questions?
relevant for understanding this paper.
Yeah, of course. The whole-
So the reduvid bugs, there are lots of different kinds.
Reduvido.
That's right. The triatomids,
Triatomids,
kissing bugs, assassin
triatomids.
bugs. There's a lot of different words for them, but they're all vincuna bugs. Those are all the same bug, okay? And they're hemipterans, and they're large. They're very large. They're 1.5 to 2 inches long. So they're quite visible. So they don't bite during the daytime. Actually, they bite during the night when you're asleep.
Fall on your face,
They
right?
fall in there. They have a very gentle approach, and then they carefully select a little zone around your eyes, usually, and then they take some blood.
How could you sleep through all this?
Well, that's the interesting.
First of all, it
part.
falls from the ceiling,
No,
right?
that's the interesting. Not always.
It's not going to fall on your face. It must fall.
No, but
on
they can
your-
fly. These things can fly.
They fly
also.
and then
They're
they
flying.
land gently on your face.
Hey, come
The
in
big,
like.
the big reason the face, right, is that's one of the things that's exposed when you're
Yeah,
Yeah,
of
that's
course.
right. So you're sleeping, and so here comes this bug, right? And the bug is absolutely filled with metacyclic trypanosomes. Okay, and that's the infective stage for this parasite, and it's in the hindgut of the organism. So it starts to take a blood meal, and it fills up so much so it swells up about 2 or 3 times its volume in size, sucking blood. And it pushes the metacyclic tryps out the back end, and it defecates onto the surface of your face. You have a delayed response to the bite. The bite itself doesn't introduce the organisms. The bug then flies off, and then you start to develop this urticaria and an itch at the site of the bite, and it forces the patient, the future patient, to start to rub their eye, and they contaminate their fingers with the feces of the bug, which has got the infectious stage, and they rub it right into the mucous membrane of their eye. And that's how this organism classically gains entrance into the body. It's a trypomastoid. It's got a flagellum. It's got a huge kinetoplast. In fact, it's the largest kinetoplast of any hemipteran. Sorry, let me back that up. It's got the largest kinetoplast of any kinetoplastidae, which includes the leishmania and the trypanosomes, okay? And this paper deals with some aspect of DNA replication. But it's also got a big nucleus, okay? So the organism then travels throughout the bloodstream until it gets to a tissue. And it doesn't matter which tissue. It's capable of penetrating lots of different kinds of cells, heart cells, nervous tissue, all kinds of things. When it penetrates the cell, it goes right into the naked cytoplasm. It doesn't make a parasitophorous vacuole like most other parasites would do. It's got a capacity to go right into the naked cytoplasm. And then it rounds up, loses its flagellum, and develops something called the amastigote stage, which is the replication stage in the mammalian host. And it replicates, and it starts to divide, and eventually it ends up killing the cell. In doing so, if that cell is contiguous with another similar cell, it can just go from one cell to the next this way. Or it can differentiate back into a non-dividing trypomastigote stage. This is a long explanation of a short life cycle.
Yeah.
So the parasite... Well, no, I want to be complete because it's important to understand that what we're going to be talking about... So the trypomastigote, which is non-dividing, has the capacity for going from tissue to tissue. So if it's in the heart, let's say, and it kills off some heart cells, and then it produces this trypomastigote stage, that can then migrate through the blood supply and end up in the mesenteric, not lymph nodes, but the myenteric plexus of the large and small intestine, where it then infects that nervous tissue and it starts to cause hollow organ disease by destroying those plexes that maintain tonus of the muscle tissue. All the while, it's dividing as the amastigote stage only. So how does the trypomastigote figure into this life cycle? Well, the reduvid bug has to suck up a lot of blood because there are very few circulating trypomastigotes. Very few. All you need is a couple. And the reduvid bug then becomes infected. And it sticks by a lectin binding type of situation to the gut tract in a specific place in the anterior portion of the gut tract of this bug, where it differentiates into the epimastigote. Then it sort of grows a while and multiplies in that position until a certain time in its life cycle says, are you getting pretty tired of being an epimastigote? How would you like to be a trypomastigote? And then the metacyclic trypomastigote stage evolves from the epimastigote stage. And I realize it's anthropomorphic.
Well, we're going to convert that anthropomorphism into science today.
Exactly. Exactly. And it also evolves around single-stranded DNA.
Well, not all of it, but
biology.
Part of it is.
Yeah.
that these transitions that you've alluded to
Yes.
in some detail. It took you half an hour to talk about.
Sorry.
I think we're done now.
We would like to know the basis, or start to understand the basis for these transitions.
Right.
And in other eukaryotes, cell cycle regulation is important. And one of the important proteins there is called replication protein A, RPA. In Drosophila, this regulates the cell cycle and therefore transformation. So they wanted to know, is there an RPA here in T. cruzi? And is it involved?
Even in Saccharomyces cerevisiae,
yes,
right?
Of course. This is a broadly distributed protein. Right. It's a single-stranded binding protein. It's important for DNA replication.
Right.
Because when, we all have double-stranded DNAs in our genome, right? In order to replicate them, the strands have to be separated so the polymerase can attach to each strand. There's bidirectional replication, but the structure, the replication structure, has to be maintained in a single-stranded form. It can't re-anneal into a double strand. So the function of the single-stranded binding protein is to bind to single strands and keep them apart.
That's interesting.
Very important.
It's like a wedge that you place between two things that want to come back together.
In fact, there is a single-stranded DNA binding protein from adenovirus, which looks like a plow.
Interesting.
It plows into the double strand and helps to push them apart, and then it remains bound. And so you keep the single. And then when you make a new strand, of course, it anneals to the template. You have double strands again. But yeah, it's a very important protein. So that's what they're studying here. RPA.
So they have three different subunits of this. Is that
this is correct? A heterotrimer, exactly. Hetero means different.
Yes.
Tri means three.
And a murmur.
Boy, we can just pull apart words by looking at their roots.
Well, it's important for some of our listeners, including me.
Are you a listener, Dickson?
I am a listener. I'm a listener.
So you have the genome sequence of T. cruzi. You can just look and say, is there a RPA?
That's right.
And
they did that.
And they found it. There is...
Sure.
And they call it TC-RPA because the Trypanosoma cruzi.
That's right. So we're talking about the nucleus now. We're not talking about the kinetoplast because that's got DNA in it also.
Nuclear DNA.
This is nuclear
right.
DNA. Okay.
There, later on, I'm going to talk a little bit about that, the localization of this.
Okay.
So, Dickson, if you just had a sequence, what could you do to see what the protein did, theoretically?
You could look for domain similarities in the gene bank.
Exactly, because you got lots
of protein bank.
You have lots of sequences in gen bank.
You got a ton of them.
So you can say, let's compare them.
I have even done that.
And if you have an RPA from many different organisms, you could say, does this look like it?
Exactly.
And in fact, not only do we have sequences, we have crystal structures, a lot of those proteins as well.
Right.
And so they did say, hey, it looks like, smells like, seems like, must be.
So another way to approach it would have been to make a probe based on the mammalian RPA1 and see if it hybridizes.
You know, that approach reflects your antiquity, Dr. Despommier.
Probably, but still.
But yes, in the days before we had genome sequences, you would do that.
If you didn't get an answer, that means that it's
But
not
nowadays
there.
we have total genomes sequence. You don't have to do that anymore. You can just look at that.
I got it.
Now, of course, would you know how to do that? That's the question. If I say to you, Dickson, go find the T. cruzi RPA, would you know how to do that?
Sure.
What would you do?
I'd get Amy and I'd say, Amy, now you're going to help me.
Well, that's one way to do it.
Probe the gene bank and
get someone else to do it, right?
Well, no, you could look it up. I'm sure it has a number, an acquisition number, and then you could go and snip out a piece of its sequence and see whether or not the same thing is present in the DNA sequence of the trypanosome.
So there are two forms of this protein, 1 and 2.
Right.
TCRPA 1 and 2.
And they're not sure they have a third trimer, are they? Because I didn't see them mention it.
No. These are two different isoforms of the trimer. That's what I'm talking about, right? Right. So then they say, it looks like a DNA binding protein. So let's actually make the protein.
Yeah.
And you can put DNA in. What do they do it in? E. coli? Do they produce it in E. coli? Probably. They can produce the protein. And they can see, does it look like a RPA of other species?
Does it look like?
Well, they do circular dichroism.
They
Dickson.
didn't. No. Where did they get all those crows from?
Should we explain what that is?
Go ahead, explain it. We tried before. We didn't have much luck.
Oh, you two tried to explain it to each other? Okay.
It's a way of looking at the spectra of the protein.
It's interesting.
The absorption.
So we have that whole thing like that light can be thought of as waves and as particles.
Mm-hmm.
And so you could think of light as oscillating up and down, sort of in one direction. That would be sort of a linear. But circular, what you do is, so there's a sort of a field to electricity. And so if you rotate this field, you're rotating the vector. And so think of the wave now, instead of oscillating up and down, of basically spiraling. And it can spiral to the right or it can spiral to the left. And by basically applying this spiraling to the right or spiraling to the left, that's your circular dichroism. You can use this, think of it as a polarization. Basically, you can shine that at your proteins, and you can look at the spectra. And then, as they're going to do later, they're going to then expose it to DNA. And if it binds to the DNA, it's going to change the circular dichroic pattern.
So with
Can I make one small
No, you can't.
correction?
No.
Oh, you make go ahead.
I would like to differentiate the difference between a spiral and a helix. And I'm not kidding, because a spiral has a single point and then it continues to expand its radius outward, whereas a helix remains the same diameter all the way through. So these are actually helices that you're talking about. Is that not correct?
Yeah. No,
okay, I just wanted to make that clear because having dealt with Trichinella spiralis all my life.
Yeah. No,
so if you can look at the CD spectra of a protein, you can tell the percentage of alpha helices and beta sheets and disordered elements.
Helices,
and then they did it with these proteins and say it looks very similar to the other RPAs that we know. And then as Daniel said, you add single-stranded DNA, it changes the CD spectra, but only TCRPA1 was able to apparently bind DNA. which they then confirmed by gel, what we used to call gel shift assays, which is really a bad name because the gel doesn't move.
Right. The band inside the gel.
The band inside the gel.
Right,
if you have a protein running on a gel,
Yeah.
and it will run at a certain place, and then if you add DNA, it will retard the migration. They're really technically electrophytic mobility shift assays, EMSAs.
EMSAs, yeah.
Right.
But the colloquial was gel shift, which was always criticizable, right? So TCRPA1 binds single-stranded DNA by both CD and gel shift assays. I'm going to use the bad word because I like it. And,
and why would they not expect it to do that? I mean, it seems pretty
the history.
straightforward at this point.
It looks like an RPA, but it doesn't bind single-stranded DNA. And that's interesting. I don't know if they expected that or if that was unexpected, but that's what they found.
Right.
And maybe it has some other function, but it happens to look like a single-stranded DNA binding protein. I just don't know.
Mm-hmm. Well, I think that's the valid sort of point. There, the suggestion is it has homology at the amino acid level to DNA binding. Let's go ahead and see if it binds or not. And here they show that the RPA1 binds, the RPA2 doesn't bind. And then they're going to go to the next step of looking at different binding motifs, different sort of sections of the amino acids, and saying which section binds to the DNA. So they're using these OB motifs, OBF1, OBF2, and OBF3.
So just to be clear, in the mammalian system, 1 and 2 do bind, but in the trypanosome system, only 1 binds.
I don't know if this... I don't know.
So there's RPA 1, 2, and 3 in the human system, and RPA 1, 2, and 3 forms a
trimer,
heterotrimer, and there are 4 OB domains in the human system. And the OB domains is the oligonucleotide binding, so OB. So there's 4 when that thing forms, the 3 get together. There are 4 places where it can bind nucleic acids, and in this, like I guess we're discovering. They're looking at what might be these different OBs, so oligonucleotide binding regions. And what do we find? We find
that they do some modeling of the structures because we know structures for other eukaryotic DNA binding proteins. We have these 3 binding domains. They take, let's model single-stranded DNA into it, and it looks like they can bind single-stranded DNA of TCRPA1 similar ways that other eukaryotes do. Yeah. And then they make truncated forms that have just OBD1 or 2, and they can only find that OBF1 only interacts with single-stranded DNA. So it's different in that sense with the...
That's sort of isolating which motif is binding, which section is binding.
Sure. And I would just raise another question then.
Yes, sir. You bet.
Which stage of the parasite did they derive their reagents from?
So they take nuclear DNA, they produce proteins in E. coli, so there's no stage involved.
All right.
Not yet, but they're going to get there.
They're going to get to that in a different way, but not yet. So the conclusion of this so far is that the DNA binding mode of these RPAs in trypanosomes may involve fewer nucleotides and can be processed in a different way. Okay, so there's a difference. Maybe that has something to do with the biology that these things can transform, but we don't know, right? Now, they next look to see what other proteins this TCRPA is interacting with. So they do immunoprecipitations with antibodies, and they find that an antibody to TCRPA2 will immunoprecipitate TCRP1.
So they're binding.
They're binding.
The 2 are interacting.
That's what is expected. That's not a surprise.
And RPA co-localized with replication sites in the nucleus, but not in the kinetoplast.
That's also not surprising.
So you think that the kinetoplast has its own DNA binding proteins that are that different?
They are.
A little surprising,
different.
Really?
Yeah. The DNA,
why is that? Because they came from bacteria, right?
Probably. the kinetoplast DNA consists of maxi and mini circles, and those are complex arrangements of DNA compared to the nucleus that's all spread out and got hemachromatin and all these other things that were the typical nucleus. They're not typical once you get inside the kinetoplast. They're very atypical for sure.
Do you think there's an RPA in the kinetoplast? Do you think they have their own special
that would be the
binding proteins? Yeah. I mean, they must, right? Because they're also going to replicate. They're going to need something to hold the
This is very true.
strands separate.
They have to.
Their replication is bizarre.
They have to have a single strand of DNA
It's
binding.
quite bizarre.
Yeah.
Sure. They also stress the cells. They induce replicative stress by treatment with a chemical, hydroxyurea. And then they look at nuclear localization. And, this stress synchronizes the cells at the G1S transition point. Right? So S is the synthetic phase, and G is gap 1.
Okay.
Right? So right before DNA synthesis, if you stress the cells, they stop. They don't go into synthesis. And then they can see foci of TCRPA 1 and 2 in these cells. And they think, because of this, that these are involved in DNA damage repair. Right. So the induction of stress and DNA damage through UV irradiation causes these proteins to arrange in a punctate pattern at the nuclear periphery. And so they think they might be involved in DNA damage, but it's very indirect, obviously. Just suggestive. They'd have to do some more experiments to figure that out. All right, now, Dickson, they move into the functional experiments.
Okay.
They do a heterozygous knockout of TCRPA2. TCRPA2. Let's go back and remind ourselves which of the two. Only TCRPA1 bound DNA. So this is the one that doesn't bind DNA.
The other one.
You want to know what that's doing, right? Because if you knock out TCRPA1, you're going to have problems replicating.
Right.
So they have obviously the technology to knock out just one copy. So a heterozygous knockout means you just knock out one of the two copies. These are diploid organisms, right? Dickson?
They are. And that's interesting, too, because it took them a long time to find the sexual stage for this parasite.
Mm-hmm.
But they did find it.
They couldn't make a homozygous null. They were not viable. So you need RPA2 in trypanosomes. They're essential. Okay, so the best they could do is a heterozygous, which reduces the level of the protein by how much, Dickson?
50%.
Exactly. 50%.
Ask me how many genome types there are in
How many virus
7.
replication types? So reduction of TCRPA2 impairs cell growth.
That's interesting, right?
All right.
But
it slows down DNA replication.
Yes.
But what you're looking for is
the money shot.
The money. Hey, this is a family podcast. It increases differentiation. Epimastigote differentiation.
Fantastic.
So, Dickson, explain to us how you assay differentiation.
Well, they're just different shapes of organisms. You can
have
tell
an in vitro
apart.
culture. How do you let them differentiate on their own?
You can do that, or you can raise tsetse flies and do it in the tsetse fly colonies.
Unlike the effect on T. cruzi proliferation, the reduction of levels of TCRPA2 increased differentiation, 3 times more metacyclic trypanosomes.
Remarkable.
Compared to wild type.
Isn't that interesting?
Why? So this is TCRPA2, where we don't know. It's not a DNA binding protein.
Yeah.
So it slows down differentiation.
Yeah, it appears to be regulating it in some way, right? So the knockout grew slower, right?
Right.
Had a problem with DNA replication, so it's obviously involved in some aspect of DNA replication. Not DNA binding, apparently, right? We found that. But now it increases differentiation. That's cool. That is really the neat finding here, I think. But we don't know why.
So then you wonder if those metacyclic trypanosomes are actually infectious.
How would you do that?
Well, you'd need a host. And T. cruzi infects almost every mammal.
We're hosts, right?
We were told we were hosts.
Yeah, someone addressed us as hosts.
But you can use any kind of mammal because...
a mouse. You can put them in a mouse. Yeah, that would be their next paper, I guess.
I believe.
Although if you were a reviewer, would you demand that to be part of this paper?
It's an easy experiment. Why didn't you do it?
They go ahead and do this, actually.
They do the experiment?
Yeah, they do this. Let's go to figure 7.
7?
So figure 7, they do these knockouts, and then the knockouts have a higher capacity of infection compared to wild type cells. It's like about twice, even more. Yeah, and that's that interesting, I think, from your life cycle, as we talked about. You have this replicative stage, and then you stop replicating and become infective. And what they're saying is exactly that. we saw from these assays that they're not replicating, and then we see in figure 7 that they have a much higher number of cells that they infect. So they're infecting twice as many cells.
So they're infecting cells in culture here.
Yeah.
Okay.
So these will get into the cells, right? So which is the form that would enter the cell?
The metacyclic trypomastigote.
Not the epimastigote,
No.
right?
The epimastigote is in the anterior portion of the gut tract of the reduvid, and the metacyclic trypomastigotes are in the posterior
The
portion.
hindgut.
Right. So the
Hind
knockout
gut.
makes them more infectious for
Yeah, so you
cells, yeah.
sort of have to make a decision, I guess, right? We'll anthropomorphize. You're either replicating or you're infecting.
Yeah.
And so if you stop replicating, you can start infecting. And this seems to affect that.
So I wonder if it knocks itself out. Knock yourself out.
Go ahead,
so to
knock
speak.
yourself out.
I wonder if that's actually what happens to make them infective, to transition from an
Maybe.
epimastigote to a metacyclic.
Maybe that's the question here.
Yeah.
How does it? How does?
But if you
this
knock it out
regulate?
in it double strands, then it can't replicate at all.
Well, it's dead. It's not. It's not infectious. It
Right.
can't grow because it's needed. All right. Without it, it
But
can't.
don't know why.
Well, I presume it's involved in DNA replication. So, yeah, but you don't. I don't think they know the answer, right? If you have a null, which that is non-infectious or not viable, I should say, you can't really figure out why, because they don't live.
But if you
know?
knock out one of them, you get a transition from an epi to a metacyclic without replication. That doesn't involve
with reduced
replication.
replicate, well, there's reduced. Remember, because there's a 50% reduction in the amount of the protein. So replication is not zero; it's lower, but somehow lower replication and higher transformation are going together.
So does this imply that the epimastigote must divide in order to differentiate?
No, it doesn't.
Because it could sit still and
Yeah,
just
right.
transform into the next stage
Yeah.
without division.
No, right. It doesn't. It's not a division. It's a transformation.
No, I'm thinking
right?
of but you think of other things like B cells and their maturation towards plasma cells, and every division cycle resembles a different mature cell until finally it is that.
That's an interesting one you bring up. People have this idea, right? As a B cell develops, it keeps dividing, but it actually is like this. It transforms, and it's not until much later that you actually get any division. So it's the same cell that is transforming.
So is the epimastigote undergoing a similar differentiation process?
Yeah, I don't know.
It looks like once they stop replicating, they differentiate. Once they differentiate, they have the ability to penetrate and infect all our cells.
But here's the conundrum. You've got this other piece that doesn't involve DNA replication, and yet you can't live without it.
Yeah.
I wonder if it's a DNA binding protein, but it's a transcriptional regulator.
Well, the fact that it binds to a DNA binder. I mean, we have a lot of situations where you have a particular protein that's the one that actually binds.
What kind of
through
motif
whatever,
does it have?
And then it binds to that. So they sort of build on each other.
I know for a while we were working in our lab on a basic helix loop helix protein, which is a transcriptional factor.
That's right.
And I wondered whether any of these fit into that category.
They didn't mention it, but there are other motifs that are not just like that could.
Yeah.
I'm just wondering if it could regulate gene expression, and that's how it could control differentiation, right?
Yeah.
But
the mystery continues.
yeah, it's an interesting story. I mean, there's a lot of basic stuff. More than half the paper is saying this is a DNA binding protein, and the other one is not.
Right.
The real interesting part is the last quarter, which I'd like to know more about. So.
But I think the challenge here is that if you can understand what allows this parasite to transform into the infective stage and somehow intervene there.
Well, that's right. They're thinking about a drug possibility here.
Yeah,
sure.
of course.
And you maybe spray the bugs, spray the reduvid bugs, right?
Or put it on cream on your face before you go to sleep. No, that's no good. You don't want to.
You don't want to.
You don't want to do preventive drugging, right?
Yeah, that would be too hard to enforce the use of.
All right. We don't have an ad, so we can go right into a case study.
Oh,
we
wow.
can do this.
Yeah.
We can do this.
We have a month off from ads.
Okay. Well, as I mentioned, I was going to continue with my theme of when the healthcare workers get sick. But I thought, you know what? Let's take a little break.
Did you see any patients in Japan? Now you're on vacation, right?
I actually was on vacation. I wanted to go visit the Parasite Museum, and I actually have a buddy who's a doctor there. I just was busy with family and holidays, so... this is actually one of the few times that I actually just focused on my family and not seen patients and gone in. Anyway, so here's our case. This is a case, 28-year-old man coming from a referral hospital up near the Thai-Burma border. And he reports fever and chills for 2 days. He's having a daily, pretty significant high fever, shaking chills. He reports that he feels poorly. He's only making a small amount of dark urine. And he says, as I mentioned, these fevers are very severe. He has severe shaking chills when they occur, but no rash, and he's having these fevers 1 time per day. It's only been 2 days, right? So, I take that with whatever. He doesn't report any diarrhea, but he has some difficulty breathing. So when he was seen initially by a local healthcare volunteer, they were kind of concerned, went to the local hospital, and at this point, he's been referred to the tertiary hospital in Bangkok. Okay. And he's up there, as we mentioned, up by the Thai-Burma border. He has an exposure history to pigs, dogs, chickens, mosquitoes, etc. He's working up in the jungle. I think it's involved in the timber industry, but it was sort of unclear what exactly he was doing up there. But he is sleeping out at night without any kind of protection. He doesn't have any prior medical problems, no past surgical history.
So sleeping out at night with no cover, right?
Exactly.
Just clothes.
Just his normal clothes that he's wearing.
Is he wearing shoes when he sleeps?
He's got sandals.
Sandals.
Very nice sandals. No, he has sandals. Nice there. He doesn't take any medications.
Is he married?
So he's not married, but his mother, father, and siblings live up there.
He lives with them?
So he lives with them. And he says that he reports being a farmer. I think we had this idea that maybe he was involved in the timber as well. No toxic habits. We believe he's HIV negative.
You believe. Okay.
We believe he's HIV negative. Okay.
Does he have sex?
He does.
He doesn't have a wife though, right?
He doesn't have a wife, but he's heterosexual.
Does he go to brothels?
Not that he reports.
Okay. But he reports being sexually active, right?
Yes.
Okay.
Say how old again?
He's 28.
28. Now, tell us a little bit about his, well, maybe you can, go ahead. I won't interrupt.
So let me go ahead. We'll do the vitals and exam, and then I'll let you guys. So when he's initially seen, he has a high fever, low blood pressure, a rapid heart rate. He's breathing rapidly. He appears acutely ill. He has scleral icterus, right? So the whites of his eyes are yellow, dry mucous membranes. His neck is supple. His lungs are clear at auscultation, but he is breathing rapidly. Heart is rapid, and he does have a 2 out of 6 systolic murmur. So we're hearing a heart murmur. His abdomen is soft but tender, mildly tender. He does have normal bowel sounds, but he has an enlarged, slightly tender liver and an enlarged spleen. And his skin, he has no rashes, but he has lots of cuts and bruises and bug bites.
Why is that? Where does he get that from?
He says from the farming, from his work, from...
So you said he's involved in the timber industry and farming. What is it? Those are two different things, aren't they? Is he farming trees?
No, I think it's a mix of things. I don't...
Oh, okay, got it.
He's probably clearing land to make room for farms, and that's part of the timber industry that he's part of. That's typical.
Yeah.
He should build a vertical farm.
And I'm going to give you some labs. Right?
Okay.
So I'm going to give you labs, and then people get to think this through and tell me what it is, okay?
All right.
Some people may feel like, oh, you're giving me too much. But you know what? I'm going to give you too much, and I expect people to figure this one out. So he's got low platelets, low hematocrit, low glucose, and we're in Thai on the Thai-Burma border, so they do a blood smear. And the blood smear is abnormal. We're seeing 5 to 10% infected red blood cells with multiple band forms.
5
These
to
are
10% infected.
Infected RBCs.
You're not going to tell us what they're infected with.
Well, people can think about that.
With what band forms?
Let me say that and he has multiple band forms. These are not white blood cell bands, but band parasitic forms in the RBCs.
Got it.
And can you tell us?
And I said 5 to 10% of his cells are infected.
And can I ask another question?
You could ask.
You
might
won't
not
give me the
answer 'cause
answer?
I'm giving a lot here.
Yeah,
so.
you're giving an awful lot. The size of the red cell that's in.
Yeah, I've
Got
given
the
you
band.
enough.
You-
That's a good question. Good question.
I know.
But remember, this could be a red herring, you know.
A red cell herring.
Yeah.
You've given a lot, actually.
And people are welcome when they write in to suggest what size they think the red cells would be and why.
Yeah, that's right. They have to justify it based on their parasitic diagnosis.
And this
Yes.
has only been happening for 2 days. Before these 2 days, he was absolutely fine, right?
Well, probably.
Hard
So
to
he
say.
says.
Hard to say.
He says it all started two days
It all
ago.
started. Coincident with any particular activity or encounter?
No, he doesn't report any coincident activities or encounters.
But he's outside and he's got lots of contact with insects, right?
Yes.
And he sleeps outside at night.
Yeah, that's the key there, isn't it, Dickson?
Well, it could be.
Maybe.
Yeah.
Could be. But remember, his liver and his spleen are enlarged.
Is he
His liver
eosinophilia? Does he
and-
have eosinophilia?
Excuse me, we're just talking now.
I know. I'm talking to you.
I don't think he told us.
No, he didn't tell us about eosinophils, but he did say both the liver and the spleen were
He
tender
did.
and enlarged,
He did
right?
say that. He did say that.
Hepatosplenomegaly.
That's what I'd call
That's
it.
the term.
That is the term.
And we're in Burma at the
Wait.
Thai border.
Right.
Mission of Burma. Remember that group? No, you don't remember.
Actually,
Yeah. It was a rock group up in Boston.
don't remember that.
Yeah.
I do not.
I bet you some of our listeners remember. Anything
Remember
else?
the B-52s.
Yeah, those are more globally well known.
They were the McNamara's favorite singing group.
Well, do you guys have any other questions about this case? You feel like you've gotten enough
What does he eat?
information?
What
Wait
does
a
he
minute.
eat?
You say he lives with his parents and his siblings.
Yes.
Yes.
How are they? They
Yeah.
good? Are they good?
How are
They're fine.
Do they sleep inside?
He sleeps with them?
So they don't have any. I should say no mosquito nets. They live in these rustic dwellings.
Rustic dwellings.
No windows, I guess, right?
There's no glass windows, but there are holes where the light comes in. And
So
the breeze
basically,
and other things.
they're all sleeping in the same situation because the inside is outside.
Well,
right?
I did mention he mentions this logging. He's off doing this timber stuff as well. So he's primarily, he says he's a farmer, but then he also does say that he's been off in the
He works two different jobs.
deeper in the jungle doing.
So when he
timber
works in this
stuff.
timber stuff, that's when he sleeps outside.
That's when he's like
When he's
sleeping
home,
outside.
he sleeps in the house. But there's no windows, there are no screens open. How is the family? Are they all okay?
Everybody else is doing okay. It's just him.
All right, family is fine. So when
he eats, he eats standard Thai fare. I think we've discussed that before.
Standard Thai fare covers a huge
jumping salad.
cuisine. No, that's Filipino.
That's Filipino.
No, actually, it's
Daniel,
big in Thailand too. I
Is
think
it
we had
really?
this discussion.
Jumping
Yeah,
in-
it's in both places.
Daniel, tell me, when he goes to do this timber stuff, what's his elevation?
He's not that high, actually.
Not very high? Okay.
Not very high.
So they're probably mosquitoes.
That's a good question.
They're mosquitoes, Dickson, right? You go too high up, there are no mosquitoes, right?
Well,
Remember the paper we did on TWiP
of course I do.
where we showed that the range of mosquitoes is going higher and higher
It is
because
because
of global
climate
warming, which does
That's right.
actually happen. It's true.
You're right.
But is it
Right.
caused by us? Are you
Yeah,
trying
it
to
is.
blame us?
It is, yeah.
Again,
I see movies where there's apocalyptic things happening to Earth,
yeah,
and it's scary.
it is.
Yeah.
It is actually.
It's very scary.
Yeah.
Yes. I see lots of frightening movies too.
There's anarchy and there's no more law and order, and these politicians don't realize what they're ridding on.
The crazy things-
Ridding on us. What's the word I'm looking for? What they are causing us to have.
They're causing us to be apoplectic.
Anyway, so
Having
he is
bad
at
dreams.
an elevation where there are mosquitoes.
Yes, that is
course.
fair.
All right.
That is fair to say.
Got it.
And I share your same concerns.
The other risks that he's got, think about where he works. I mean, Think of all the wild animals that are in those areas.
What kind of, tell us, Dickson, what kind?
Tigers.
Tigers? Really?
There's lots of tigers.
Well, he hasn't.
Not jaguars, but panthers. They have panthers there. Leopards.
Lots of cobra. Lots of snakes.
They have lots of venomous spiders of all kinds, and it's not a pleasant environment.
It's a dangerous job.
It is.
It's like the Alaskan king crab harvesters, right? Most dangerous job in the world.
Well, I wouldn't say that, but it's one of the most dangerous at times.
Should I mention that he's been bitten recently by a snake? No, just joking. That's the last thing he needs. That too.
Yeah, right.
He has no snake bites, right?
No snake bites.
Speaking of
nor any tiger bites.
you would certainly know that one.
Yes, Dick. Sorry to interrupt.
No, you're not. I was going to mention.
I'm not sorry.
No, you're not.
Okay.
I was going to mention an unusual event of a good nature that happened around here about 2 or 3 days ago. A humpback whale went up the East River.
So you're going to say he went up the east side to get something to eat.
It went up the East River.
Yeah.
Is that bad?
And out into Long Island Sound, I think.
Yeah, they actually migrate through the Long Island Sound.
It's quite remarkable.
And, the marine ecosystem's really coming back, and there's whales off Breezy Point now. And
you don't sail in the Sound, right?
I do.
He certainly does.
Yeah.
I thought you sailed on the ocean side.
No.
No, on the northern side, so I'm mostly in the Sound.
So have you ever seen a whale?
Yes.
Amazing.
So, Dickson, why is this it's really cool that the whale did this, but is this an unusual occurrence?
It is.
Yeah.
We've actually had a whale come right up to the George Washington Bridge once.
Did you see it?
Not too long ago.
We
I
had the
didn't
issue
see
with
it.
the belugas last summer where we couldn't get to the dock because there were all these belugas between us and the dock, and we're like, what's that?
Well, that's another sign that might not be so great because belugas love very cold water, and they're being chased out of their zone by this warming trend that we're experiencing.
Interesting. Was that in the news, this whale?
Yeah, it was. You could find it.
Hey, Dickson, do you have a superhero for today?
Oh, not today.
You forgot, huh?
No, I didn't forget. No, I did not forget.
You don't want to do it every time? You want to do it every other time?
I could do a brief one. How's that? I will give you
if you're not ready, it's fine.
We'll do a reprise. We'll do it next time.
Next time is fine.
Superhero.
All right, because I have completed... I guess I should preview this in TWiP because Parasites Without Borders, as you know, is our group. Daniel and I are in cahoots here, and we've produced the 6th edition
with Chuck Knirsch.
With Chuck Knirsch and 2 other people too, Bob Guads and Peter Hotez. And the book has come out in hard copy now, and it turns out that there are 28 blank pages in this book. And one would say, "My gosh, how could you have a book with 28 blank pages?" So we've looked at other books and said, my goodness, there are lots of blank pages in lots of books.
I was okay with the blank pages.
He was okay with the blank pages. And I was a little
problems.
bit upset because I realized that here's a missed opportunity. But, we only produced 1,000 of these books. So I took it upon myself, and now I had the approval of the board of directors, which is the other 2 people.
And the majority of them are already actually sold.
That's right. So what we're doing is filling up the blank pages with parasite superheroes. Each page will get a picture, a portrait picture, and a brief, concise biography of the individual as to the contribution that they made towards tropical medicine or parasitology.
You're talking about the next printing, right?
Yes, which is going to happen really soon.
All right.
So we did Sir Patrick Manson last time.
Yeah.
And so and this time, for the next time, I should say, I'll give you a preview of this. I want to do this guy, Francesco Redi. Francesco Redi is a very interesting early parasitologist.
Right. Whenever you're ready.
And that was very good. I like that. That's very good. That's excellent.
Thank you very
so
much.
next time we'll be ready for Redi.
Okay. We have some email.
Daniel,
can you take that first one?
Yes.
This was actually a response to the last case that came in a bit late.
He was just slow. Yosef writes, Dear TWiPlets, due to the vague nature of the symptoms and lack of buzzwords in this case, I have gone with several worms in order of what I think are most to least likely. And this is the mango eater. My most likely worm would be Strongyloides stercoralis. This worm is absolutely prominent in DR, can cause a diarrheal illness. The worm may be obtained from the soil and burrows through the foot of the host. It then travels to the lung capillaries, goes up the trachea to finally be swallowed in the nice, moist, warm, comfy GI tract. If the host is immunocompromised, then the worm may disseminate and form a thumbprint rash. I believe that Strongyloides could be the appropriate size, and so fits the case most well. I was going to just throw in, there's two classic strongyloides rash. There's the larva curans, sort of the running larva, and these are running like a centimeter an hour. And then there's the thumbprint peri-umbilical purpura rash, which is, it really looks like people have made thumbprints with purpura. So they're actually slightly different. One is you're seeing the tracks of the super speedy strongyloides migration. The other is you're seeing this inflammatory purpuric reaction. So just a lot of people sort of lump those together, but because
And it had another name in the old days, shoe button.
Okay.
Shoe button.
Does anyone know what a shoe button? I don't even know what a shoe button is.
It was like
none of my shoes have buttons.
A large, but in the old days, you see a large lobate thing with a little narrow neck, and then the intestine has got lots of these where the worms are actually found. So you can see it on radiology pictures of heavy infections with strongyloides.
Wow. Hookworms are also on the differential in the Cater americanus or Ancylostoma duodenale. Infections may lead to diarrhea, but also usually eventually to an iron deficiency anemia. Heavy infections may also present with respiratory symptoms due to the migration of the worms through the lungs on their way to the GI tract. The patient does admit to walking barefoot, which would provide the right risk factor for this infection. Furthermore, the adult worms may be around a centimeter as reported. The reason this diagnosis may not fit is that there doesn't seem to be any symptoms of iron deficiency anemia to clinch the diagnosis. I would look at the patient's feet to see if I could see any serpiginous lesions or rashes. Next is Trichurus Tricura, also known as whipworm. These worms may cause an inflammatory diarrhea that can be what our patient is experiencing. It is typically tropical, but can be found worldwide. The most disturbing symptoms that may follow are the tenesmus, feeling or inclination that one needs to empty your bowels, and eventually rectal prolapse. Neither of these symptoms are things that our patient is presenting with, thankfully. Furthermore, the adult worm is usually 3 to 5 centimeters in length and not the 1 centimeter reported within the stool. Anaerobius vermicularis is also around a centimeter in length, but the symptomatology does not match. It usually does not cause diarrhea and instead leads to an itchy behind. While I await ova and parasites analysis in the stool samples, I would start ivermectin as treatment since it should be effective on most of these worms. Sincerely, Yosef Davidoff. And P.S. I had heard before that a disseminated worm infection could lead to strep bovis seeding out of the GI tract and being found in the blood or CSF. I haven't found any evidence to suggest this, but is this something you have heard about? And if so, should all patients positive for strep bovis get a colonoscopy and an ONP? PPS. Thanks again for the textbooks to our medical school. So what will I say? I guess I'll answer this thing about the disseminated worm infection lead to strep bovis seeding. So with strongyloides, we'll often see gram-negative bacteremia and even a gram-negative meningitis. And that's because basically these are fecal-filled animals that are migrating around. And so when they get into our CSF or anywhere else, they defecate, and out comes feces with E. coli, other enteric pathogens. Strep bovis is actually a particular pathogen that is associated with colonic malignancies. So actually, as a general rule, and it's actually amazing, I think 10% of people with strep bovis bacteremia in some of the studies in that range will have a colonic malignancy. So if you do get a strep bovis bacteremia, I would recommend that the person undergo a colonoscopy. The ONP, I wouldn't necessarily think with strep bovis, but gram-negative meningitis, then I would worry about strongyloides.
Oh.
All right. Dickson, can you take Haley's, please?
Sure. Haley writes, Dear TWiP doctors, I work as a food safety and quality specialist at a produce company. So your case presentation of the woman with intestinal myiasis from fruit fly larvae was the talk of our office this morning, or rather it was me talking, making everyone else uncomfortable. Not only do we love mangoes in our office, but as a health-conscious individuals, we eat the skin for an extra dose of fiber and for its various phenolic compounds. If the urushiol had any effects on us, we are as yet unaware of them. I'd like to think that I'm inducing immunotolerance to the substance by introducing small quantities of it to the lymphocytes in my Pyrus patches. So I might have less of a reaction even if I encounter poison ivy on a hike, but maybe that's just wishful thinking. Some of your listeners might be relieved to know that mangoes imported to the United States are subject to various phytosanitary treatments before they are allowed to enter the U.S., and many of these specifically are designed to kill fruit fly larvae. Hot water treatments of 115 to 118 degrees F are common for different durations depending on fruit weight and cultivar. Ionizing radiation doses are also used, minimum levels differing based on which pests are endemic to the exporting country. Both treatments are done to protect different crops in the U.S. from pests of economic significance, of course, but hopefully this may also reduce the pests of clinical significance as well, although I'm not sure what levels would be required to kill larvae of the false stable fly or others capable of causing myiasis. Which treatments are required for which country exporting the fruit is determined by the APHIS, the Animal and Plant Health Inspection Service. Anyone interested in this can look up APHIS's treatment manual, which you can download online. See the link below. However, since parasites don't have borders, these treatments wouldn't affect fruit flies that would begin their infestations after the fruit has been imported. One case study I found online was a 12-month-old girl in Washington, D.C., who had developed myiasis after being fed fly-ridden bananas, link below. Another I found was in two children whose father ran a compost business. It seems to me like the biggest risk in the U.S. is to, sadly, the poor and or desperate individuals forced to eat food that probably shouldn't be eaten anyway, or to children whose parents do not keep sanitary households. I have a question for you, gentlemen. My mother watches Hoarders, a show about individuals who suffer from compulsive hoarding, a form of OCD that sometimes causes them to keep things that are rotting or unsanitary. Every time I see the show, I think that people with compulsive hoarding must get all sorts of random diseases that your average doctor would not expect. Do you know of any parasitic diseases that compulsive hoarders might be at risk for? Is anyone investigating this? Can you, or do you think if a doctor diagnoses a child with myiasis that the doctor has an obligation to ask about the sanitary conditions of the household and should maybe contact a social worker? I'd just like your opinions. And she gives 3 links down over here, and she says, thanks so much. Daniel, any thoughts?
Well, the biggest issue is if the people are hoarding food type objects, right? You know, anything that could be a source for something to develop.
Well, these are cat and dog hoarders mostly, or geese or ducks.
Yeah,
They're animals.
Yeah.
So cats,
They hoard animals?
yeah, they do. They've got they open a door and there's 5,000 cats inside.
Yeah.
Oh my gosh.
Yeah, so I would think of toxoplasmosis.
Yeah, well, toxo definitely, and that's well described, I think, among the cat hoarders.
That's right.
And you know, and you love the idea. Everyone, you know, they don't mind all the cats in the cat urine because they're so heavily infected with toxo that they destroyed that aversion part of their brain. I don't know if that's true.
No, it's not true.
But you know, if you're going to have dogs or cats, it doesn't even matter how many you have. You should deworm them. Actually this last year there was an entry in the it was the infectious disease fellows contest, and it was just the amount of people in New York City that don't deworm and the amount of dog and cat parasites in our parks.
Oh yeah,
I mean, it's just people don't deworm their animals. So,
but it requires constant attention, and these people don't seem to have a tendency to look after their pets. They just want to collect them.
Yeah, there was a point in time, right, when you would sign up and they would call you once a month, like, you know, and you have your thing on the fridge. You'd put, you know, that you dewormed your animal that month, but I don't know what that all is. You know, maybe it wasn't as profitable as it seemed.
Yeah. Depends on what you're hoarding. If you're hoarding spitting cobras, I think you're in big trouble. But you know, it depends on what the animal is.
Is it hoarding if you just have one tiger? So Anthony writes, I don't think that leeches have been featured on TWiP. Leeches are still used in medicine. He gives us a link here. It appears that leeches can be disease vectors. And so what do we worry about? When we get our leeches, we get special medical grade Aeromonas free leeches.
Yeah, right.
We do. And they're helpful for some things. Let's say you chop a finger off, you sew it back on, you want to keep the blood supply. You'll elevate the hand, you'll put a leech on the tip of the finger, and it'll suck the blood through.
Wow.
Sometimes plastic surgery, right? Sort of, you know, when you're trying to keep blood supply through something. But what would happen early on is the leeches, if you're not careful, could have pathogens. And so you could actually introduce an infection. So it's important to have these medically grade pathogen minimized, I'll say.
The moment leech is mentioned, I always think of the scene from the
African Queen. Me too.
And Humphrey Bogart tugging the boat, and Katharine Hepburn sitting on the desk yelling.
Yeah.
And then he emerges with all these black leeches all over him. That film, by the way, that scene was filmed in London.
Do they have leeches in London?
No, they had to fake the whole thing.
I get it, Dickson.
The reason why they had to film it in London was because they were filming on Lake Victoria, and it's got tons of schistosomiasis, and they wouldn't want to risk the actors in that situation.
Of course. He sends a link to an article from 1957 from your buddy Robert Shope.
There you go.
The leech as a potential virus reservoir.
Well, there are lots of leeches that live in water that transmit trypanosomes, believe it or not, to fish. So there are leech vectors, but not for human disease as far as I know.
So, Daniel, if you go past the two abstracts, there's another comment from him below.
Is this the tick longison implicated in the arthropod transmission of Toxoplasma gondii? And I don't remember arthropod vectors of Toxoplasma gondii have been discussed on TWiP. And he's saying here it appears that the ticks can carry the organism. I don't know if you took a look at that. This would be new to me, actually. I'm not familiar.
This would be very, very new to me also.
Do you think
this might have been
cat fleas could transmit toxo, Dickson?
No.
No?
No.
There's no evidence that you are aware of?
None whatsoever.
He sent a link to an article in the Journal of Veterinary Science and Technology, which is from Japan. There you go. Let's see what the...
It says implicated.
Yeah,
the word implicated always implicates, always indicates to me that it hasn't yet been definitively shown, but there's strong evidence that this might be important.
So what they do is they inject ticks with T. gondii tachyzoites and then explore whether these undergo... transformation.
Yeah.
And since there's no abstract, let's see what the results are. I can scan all the way to the bottom here. They found a gene in the tick, but that doesn't really... They say this 10 days after injection, they were able to PCR amplify a gene, suggesting they can harbor the parasites. I think it's not very strong evidence.
This is where a microscope would really be important.
Do you think so?
Yeah,
maybe you should give them one, Dickson.
I do.
Our last email is from Eugene, who writes, Gentlemen, I heard a review of a book, The Lost City of the Monkey God by Douglas Preston. The book describes the location and exploration of a pre-Columbian city in Honduras. Preston was among those involved in the exploration. He described the area around the city as having a particularly high density of dangerous arthropods. Apparently, all of the members of the expedition came down with a form of leishmaniasis. I've put the book on my short list and we'll get to it soon, but I thought you might be interested in reading it, as the author implies that there was something peculiar to the area which made it far more dangerous than other pre-Columbian sites. Have either of you heard of this book?
Nope.
I have not.
Have you heard of Honduras?
Yes.
Did you know they had leishmaniasis in Honduras?
Yes, I do know that.
Transmitted by...
Leishmania. That's right.
You've been to Honduras, I presume, right?
I have not, actually.
You have not?
No.
Is it more dangerous than other areas?
It is an extremely dangerous place at the moment. Yeah.
Why is that?
A lot of violence, a lot of...
not for parasite.
A lot of politics.
Not for parasite reasons.
Unfavorable politics.
You know, that's the interesting thing. When I go to these places, people always worry, going to be careful. The biggest thing I worry about, actually, is the violence.
Yeah.
High-speed lead, that's why.
People are more violent than parasites, aren't they, Dickson?
They tend to be.
High-speed lead?
High-speed lead.
Well, that will do it, gentlemen, for TWiP 124. On the first week of the year, you can find TWiP at iTunes, microbe.tv/TWiP, but most of you get TWiP on your smartphone, in which case you have a favorite podcatcher app, and then you just tell it to get TWiP. You subscribe to it. You get it free every time we release it. consider becoming a patron of TWiP and all the shows that we do. You can find all the different ways to help us out at microbe.tv/contribute. Please send us your questions, comments, and especially send us your case guesses to TWiP at microbe.tv. Dickson Despommier is at parasiteswithoutborders.com, also at Trichurus.org and thelivingriver.org. Dickson, Thank you and Happy New Year.
Same to you, Vincent, and happy birthday again.
Thank you very much. Daniel Griffin is at Columbia University Medical Center. You can also find him at parasiteswithoutborders.com. Thank you, Daniel, and Happy New Year to you as well.
Oh, a pleasure as always.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. Find his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.